Okay, here we are for part 3 of the urinary system and the functioning of our nephron. And so we've gone from filtration in the glomerulus to the Bowman's capsule. Then we went to reabsorption in the proximal convoluted tubule. We reabsorbed sodium, water followed. We reabsorbed glucose, but also lots of other things get reabsorbed: amino acids, urea, vitamin C, lots of stuff. Okay, but I'll only hold you accountable for those 3. So the next portion now of our nephron then is the loop of Henle. Remember, so here's the loop of Henle. And the loop of Henle's job is to reabsorb more water. Okay, so this happens automatically. The PCT actually absorbs the most water, then the loop of Henle. Okay, and we automatically do this. Otherwise, remember I said we make about 180 liters. We filter about 180 liters of fluid a day. You don't urinate 180 liters. That's, think of those 2-liter bottles of soda. If you had 90 of those, that's a lot, right? So we reabsorb most of what we filter, and we do it automatically at the PCT and the loop of Henle. So let's see how the loop of Henle does that. Remember I told you there are 2 limbs. Okay, here's the PCT coming down into the loop. This is the descending limb, and the descending limb is highly permeable to water. So water can move out of here, okay, but it's not permeable to salt, sodium chloride. The ascending limb, let me turn my laser on. The ascending limb here is permeable to salt, but it's not permeable to water. Okay, so when I say it's permeable to salt, it's going to take salt out of the tubule from the filtrate and put it into the interstitial fluid. Okay, water won't be able to follow from here, but we are setting up a concentration gradient because water from the descending limb, this limb is going to pass through this hypertonic environment, and we said this side is permeable, so water is going to move out. Okay, so this setup between the ascending limb and the descending limb, it's called a countercurrent flow. Water flowing down, right, is opposite of the direction water flowing up the ascending limb. Okay, now imagine, as filtrate is moving down and losing water, it's going to become more concentrated. So if we didn't have this countercurrent flow, we would get to a point where we wouldn't be able to reabsorb any more water. But with the countercurrent flow, and as this fluid inside here becomes more concentrated, we're going to pump the salt out, right? And so the fluid's going to become less concentrated as it moves up, okay? Because here we're losing water, so it becomes more concentrated. Here we're losing salt, and it becomes less concentrated, okay? So this is going to be called a countercurrent flow, and it sets up a vertical osmotic gradient that gets established by this countercurrent flow. Okay, so at this moment, I want you to just remember that the descending limb is permeable to water, the ascending limb is permeable to salt. Okay, now let's look at an overall kidney. Okay, if I can advance. There we go. Here's a kidney. Okay, and so we usually rotate the kidney like 90 degrees, right? But what we're doing is we're looking at here's the cortex, the interstitial fluid, and this is the interstitial fluid in the medulla. Okay, so the osmotic, the osmolarity of the interstitial fluid is equal to plasma. Remember, our osmolarity of plasma is 300. And so it equilibrates in the cortex, right? But also, so if the osmolarity in our plasma is 300, it gets filtered. So the filtrate coming through the Bowman's capsule and PCT is also 300. Right? Then as it starts to pass through the medulla is saltier. And that's because we're actively transporting salt out of the filtrate into the medulla. And it's going to get higher and higher. Okay, so in the medulla, the osmolarity increases progressively, and the loops, remember, project deeper into the medulla. So therefore, the longer loops of Henle, the more water gets absorbed because it's coming across a steeper hypertonic gradient, right? And again, so this is how we establish this steeper gradient is by something called the countercurrent multiplication, okay? And so that's what the next slide shows us. Okay, so this is like the loop of Henle at a moment in time. So one thing to remember, that fluid is constantly flowing, but we're gonna like do a stop motion view of that. And this is like if you're a baby and this is the very first time your kidneys start working. Okay, and so in your lifetime, your kidneys maintain this. All right, so again, fluids coming out of the PCT, and the filtrate inside of the tubule has an osmolarity of 300, right? And so this is permeable. The interstitial fluid is also 300. It equilibrates, okay? So what happens is as this fluid fills the tubule and it's all 300, remember the ascending loop of Henle actively pumps sodium out. It expends energy to do it. It uses ATP. It's a primary pump. So it's actively pumping sodium out. And it does so, these pumps do this at a rate so that it produces a concentration gradient, a difference between 200 from the inside of the ascending limb to the interstitial fluid. So we have 200 here and 400 here. Remember though that when sodium actively gets pumped out, chloride follows passively. So this is salty. Okay, so when the ascending limb does that, and the descending limb is permeable to water, right? So now this was 300. So the 300 is in a hypertonic, surrounded by a hypertonic solution of 400. So water moves out through passively until it equilibrates to 400. Okay, all the way down. Well, remember this fluid is constantly moving, so let's advance it. So here comes the fluid from above. Whoop, it moves down. Okay, so that means this 400 that was on this side moves to this side, and you can see the upper portion, it pushed up the 200, and so now there's some 200 up here. So our sodium potassium pumps keep running, and that's what shows here. And again, they're able now to produce a gradient difference of 200, right? But this was 400, and this now is 200, or I'm sorry, this equilibrated. Whoop. Right? And so as it did that and it created this difference, right? This now becomes 350 to 150. So the fluid coming down here was 300. It equilibrates, right, till it's isotonic with this fluid. Down here, we had a difference, right? We pushed 400 around and we start to pump sodium out. And so that means we added 100 into the interstitial fluid and we subtracted 100. So that's a gradient of 200, right? And so now the fluid that got pushed down here, that was 400. Now it's at a steeper gradient. It was equilibrated here, but now this became saltier, so water can still passively move out until it equilibrates. And we keep doing that. This has the ability to keep doing that at increments of 200 until the actual osmolarity of the medulla goes from about 300 to 1200. Okay. So basically what we've done here, okay, to understand this countercurrent multiplication, basically what we've done is every time fluid now comes from the proximal tubule at 300, it's going to come across 300. Oh, that's isotonic, no water movement. But as it moves down, it comes across this 450. So water moves out until the inside of the fluid, the filtrate, is 450. Okay. But as it moves down, now the area outside is 600, so water can move out until it's 600. And as it moves down, the outside now is 750. So we're getting a continuous movement of water outside of the loop of Henle. That's reabsorption. So we consistently reabsorb water. That's where the water's coming from. Remember, the peritubular capillaries surround it, so the capillaries pick up the water. Okay? Countercurrent multiplication, it can be confusing. I put a video on Canvas. Please watch the video. Okay? And you can rewind this one, and I might have a written explanation I think might be able to help you. I'll post that on Canvas too. Okay, so countercurrent multiplication. It's the system that sets up a steep hypertonic gradient in the medulla so that as fluid comes from the proximal tubule, we continuously lose water from the descending limb so we can reabsorb it. As the filtrate moves up the ascending limb, it becomes less concentrated because we're pumping salt out, right? And that helps to maintain the concentration gradient between the loop and the interstitial fluid to keep water reabsorbed. All righty, I know it's confusing. Got to watch it again and again. Watch the video on Canvas, and that should help you out. Okay, we'll continue on. We're out of our loop of Henle. The loop of Henle, its job is to reabsorb water. Okay, the sodium kind of stays in the medulla. Alrighty, and so, but that may help if you look at this picture. Here's the loop of Henle. This whole area becomes salty, has this concentration gradient. So later on, in just a little bit, we're going to talk about the collecting tubule. And this concentration gradient may help us there. So far we had this filtration that we don't control at the Bowman's capsule. We have automatic reabsorption at the proximal tubule. We have automatic reabsorption of water at the loop of Henle. Now at the distal tubule and the collecting duct, we're going to get variable reabsorption, and we're also going to see secretion. Variable means that this is homeostatically controlled. The filtrate that leaves the loop of Henle, right, and gets into the distal tubule, the body now can sense, well, what is the level of water, what is the level of sodium in our body, and do we need to reabsorb more water or less water? Do we need to reabsorb sodium? All right, so we can do that variably as the body needs. And so we're going to do that by signaling it with a hormone, right? So at the distal convoluted tubule, right, here we go, ascending limb. Now we're at the distal convoluted tubule. So this is up in the cortex, so it's not under the influence of the salty hypertonic medulla. Okay, so it's up here in the cortex. We're going to get variable water reabsorption, right? So the brain determines, do we need more water? If it does, then the brain releases, the hypothalamus releases antidiuretic hormone, right? Or the hypothalamic stimulates the pituitary gland to release antidiuretic hormone. Okay. And we'll talk more about that when we get into the endocrine system. But the target cells of this hormone are the cells of the DCT and the collecting duct. Okay, so it will bind to the receptors on those cells, and basically what it does is it inserts little pores called aquaporins, or little channels called aquaporins, which makes this more permeable to water. So it says here the DCT and the collecting tubules are impermeable to water unless the antidiuretic hormone is present. Okay, so again, oh, I had it written there. It's produced by the hypothalamus. It gets stored by the posterior pituitary gland. So we'll come back to this when we do endocrine system. Okay, and so it says works on the tubules through a cyclic AMP mechanism. So we're going to zoom in on a tubule or on a cell in the DCT, or the collecting duct. So again, here's the lumen, here's the filtrate, Okay. Here's the cell. Here's the basal side of the cell. It's got a receptor. Haha, receptor. Because antidiuretic hormone travels in the blood. It's also called vasopressin. Same thing, vasopressin, antidiuretic hormone. It has other functions in other areas of the body. It can affect blood pressure, but in the kidney, we call it antidiuretic hormone because a diuretic makes you urinate more water leaves. Antidiuretic means you're gonna reabsorb the water and not lose it through the urine. So the brain released ADH, it's traveling in the blood, it binds to the receptor. When it does, that sets up a cyclic AMP cascade, all right, which causes the DNA to put together these protein transporters called aquaporins. Okay, aquaporin is a water channel. It inserts them into the apical lumen, luminal membrane. So now these are just channels that'll let water to leak through. It just becomes leaky. So remember, outside it's a little more, really happens a lot in the collecting duct because the collecting duct goes through the medulla. So remember that it's pretty hypertonic out here. Water moves through. The blood, it's also going to be hypertonic. So it's all simple diffusion or facilitated diffusion on this side. No energy involved. The energy comes from the working of the hormone to start the cyclic AMP cascade. Okay, so that's the action of ADH, right? Reabsorbing water at the distal tubule and at the collecting duct. Okay. Yes, I'm doing that. Okay. I was also thinking, but there's aldosterone. I didn't talk about that. That's coming up next. There we go. So I'm sorry for flipping back and forth. I just put this table together so you can kind of keep track of what's been reabsorbed, how much sodium, and what its role of sodium. So we keep reabsorbing sodium, right, along the way. And so at the proximal tubule, 67% of the sodium is reabsorbed. And remember, that helps reabsorb glucose. It also helps amino acids. I didn't go over that in detail, but it does. Remember, sodium chloride always follows. Water then follows. Urea is going to also follow. At the ascending loop of Henle, we reabsorb about 25% of sodium, and that helps to also reabsorb water. Okay. Then at the distal and collecting tubules, we can also reabsorb sodium, about 8%, but it's variable. This one's going to be subject to hormonal control, a different hormone. The one we just talked about is only for water. There was nothing to do with sodium there, right? But we can also control the variable reabsorption of sodium through another hormone. And that hormone is aldosterone. And aldosterone has to get signaled to be released by a process called the RAS, the renin-angiotensin-aldosterone system. Okay, so again, this is a process that triggers the reabsorption of sodium, and this happens at the distal convoluted tubule. Okay, this is the most important and best known hormonal system involved in regulating sodium. Okay, it's activated by the release of renin from those granular cells. Here we are back at that juxtaglomerular apparatus. Remember, our DCT crosses in between the afferent and efferent arterioles. So the portion of the epithelial cells that are kind of sandwiched in between from the DCT, right? These were called the macula densa. But then I said there were specialized cells at the afferent arteriole, the smooth muscle cells. These are called granular cells. Granular cells can release renin. Okay, this is a hormone that starts this whole process. Okay, so the granular cells themselves can act as baroreceptors. They get sensitive to pressure changes in the afferent arteriole. Okay, so if there's a fall in blood pressure, the granule cells, that BP, the baroreceptor senses that fall in blood pressure, and it causes the granular cells to secrete more renin. That's one pathway. The other thing that can happen, again, if there's a fall in blood pressure, that means there's going to be a lower GFR. Okay? So that means there's less filtrate coming through, right? And so as the filtrate gets to the DCT and passes by these macula densa cells, they sense the fall in sodium chloride because less GFR means less filtrate, means less sodium chloride. So these sense that. Okay, so again, that triggers the granular cells to release renin. Okay, this could also be innervated by the sympathetic system, but we're not going to worry about that right now. So now there's renin flowing through the bloodstream. Okay, let's go on to the next picture so you can see this. Okay, so renin got released. Renin is now in the bloodstream. Okay, renin comes across a protein that's in the blood. It just stays in the blood by the liver, makes this protein. It's called angiotensinogen. There it is, O-G-E-N. Angiotensinogen. That means it's an inactive form. What activates it? Renin. Right? So when the granular cells sense there's a lower blood pressure, or the macula densa cells sense that there's less sodium salt coming through the tube, then it signals those granular cells to release renin. Renin's now in the general bloodstream, and it converts angiotensinogen into angiotensin 1. Now that's coursing through your blood. As that gets around the capillaries in your lungs, it meets up with an enzyme. There's an enzyme that seems to hang out in the capillaries in the lungs. I'm not sure if anybody knows why. Okay, it's called angiotensin converting enzyme. Angiotensin converting enzyme. That enzyme converts angiotensin 1 into angiotensin 2. Right? Angiotensin 2 now courses through your bloodstream. And as it gets by the adrenal cortex, so remember the adrenal gland sits on top of your kidney. As it gets to the adrenal cortex, the outer edge, that stimulates the cells there to release aldosterone. Aldosterone is a hormone now that acts on, we said, the DCT of the nephron. And that causes the DCT of the nephron to insert more sodium potassium pumps. So therefore, more sodium gets pumped out of the lumen of the nephron into the interstitial fluid, and then therefore into the capillary, and we reabsorb sodium. Okay, so we just reabsorb sodium. Water might follow. Okay, so that increases the reabsorption of water as well, not as much as antidiuretic hormone though. So here's the thing. We have to be so careful at regulating our salt levels that we need this long process of one inactive thing to activate another inactive thing, to activate another inactive thing, to activate the release of aldosterone. Okay? So you need to know this process. Okay, it's called RAS, right? The renin angiotensin aldosterone system. Okay. Yep, system. I forgot system for a minute. Okay, I put a video on Canvas for you to help step you through that. Okay, so the question might be, there's variable reabsorption of sodium at the distal convoluted tubule. How do we initiate the reabsorption of sodium at the distal tubule? So you need to go through what happens at the juxtaglomerular apparatus. Macula densa can sense the lowering of salt levels in the tubule because GFR went down. The granular cells are baroreceptors. They can sense a lower blood pressure, right, of the afferent arterial. So both of those cause the granular cells to release renin. right? Renin now activates angiotensinogen that came from the liver. That's just automatically in the blood. It's a plasma protein. So it activates it to angiotensin 1. That comes across the enzyme in the capillaries around the lungs, converts it, angiotensin converting enzyme, converts it to angiotensin 2. That causes the adrenal cortex to release aldosterone. That signals the collecting or the distal tubule to insert more sodium potassium pumps. So we reabsorb sodium. Okay, you can see other passages that happens, but stick with that one. All right, so here's another way of looking at that: homeostatically controlled sodium, right? You can step through this flow chart. Low sodium intake, it's going to be low in the plasma. The hypothalamus is going to cause the posterior pituitary to stop releasing antidiuretic hormone, right? And so it's going to, to stop releasing antidiuretic hormone so that we stop reabsorbing water. That increases our urine. So it keeps the water in the nephron so that we make more urine. Okay, that lowers our blood volume. Okay. If we want to retain sodium in the blood, right, or we go back around here, our blood volume's low now. We keep going. Blood volume's low now. That tells the juxtaglomerular apparatus, hey, blood pressure went down. Remember, when blood volume's low, blood pressure's low. Okay, so that tells the juxtaglomerular apparatus, those granular cells, to release renin. Skipping some steps here, but you need to know those steps. We get angiotensin 2 at the adrenal cortex to release aldosterone. We reabsorb sodium. Sodium increases in the blood. Okay, so that's that negative feedback loop on what initiated it was not getting enough sodium. All righty. What else do we have going on? There's other things. We can inhibit sodium reabsorption, right? And the heart can actually do that. Okay, so if the, remember, if we get too much sodium in our blood, that increases pressure, right? Because it becomes more hypertonic. We absorb more water from our interstitial fluid, so that's more blood volume, so it's higher pressure. So this is why we have to keep low sodium diets, right? So more blood, more venous return means that the atrium stretches. That stretch causes the release of a hormone called atrial natriuretic peptide, AMP, right? So that ends up getting into the blood, and that can cause vasodilation to decrease your blood pressure, but it also inhibits, this is the main function, inhibits the reabsorption of sodium at the distal part of the nephron, right? The distal tubule. So that's variable reabsorption. We just went over what increases the reabsorption. Atrial natriuretic peptide can decrease the reabsorption. Okay. One more function, and then we're almost done here. We've got a few more slides to go. Remember, I mentioned the third function at the nephron is secretion. So remember that we filter things here at the glomerulus, but we don't filter everything. Some goes by. So there might be some stuff as it's leaving the peritubular capillary that we still need to get out of the blood. And that's mainly potassium. Remember, we want to keep blood potassium low because especially in our neurons, when we open a channel, we want potassium to move out. So this also helps to maintain a gradient to keep it low outside of the cell. So if we didn't filter enough here, we have a chance to secrete it, to take it out of the blood and put it into the nephron. And that happens at the distal tubule as well. Okay. And so here's the distal tubule again, right? Here's blood coming through our peritubular capillary. It has potassium. Hey, sodium potassium pump. Woohoo! So we can pump potassium into the cell that's against its concentration gradient. So we need the pump, but then it's lower in the filtrate in the lumen of the tubule. So then it's facilitated passive diffusion. Okay, so just remember that about 98% of the potassium is in the intracellular fluid, right, due to sodium potassium pumps. So small changes in extracellular fluid can have a large effect on plasma concentrations, and that could affect membrane excitability. So that's why we really um, make sure that we keep it low in the plasma. Okay, so again, this is secretion. We moved from the plasma into the tubule, and this is happening at the distal tubule. Alrighty, I'm gonna leave it there, except one thing that I'll say: if the blood is too acidic, it's got too many hydrogen ions, and this means it's overcoming the buffer of bicarbonate. Okay, these pumps will actually preferentially pump hydrogen ions instead of potassium ions. So that's like an extreme when the blood becomes really acidic. Okay, what if our kidneys don't function, right? You go into renal failure. Okay, I think this is I got this from the internet, so hemodialysis, this is probably a European spelling. You can get rid of that A, right? You have to filter the blood. I mentioned we have 2 kidneys, one works, 2 is better, but 1 works. But if neither kidneys function, you have to do dialysis, okay? And any type of renal failure kind of results in the same thing. You get toxicity of the blood, and that can affect other organs. So when the urinary system fails, the renal system, the kidney, you can quickly have other organ failure. So, so you have to have dialysis, and so there are different types of, this is a blood dialysis where you're actually taking the blood out, right, passing it through a filter outside of the body and putting the clean blood back in, right? This could take anywhere from like 4 to 8 hours, and people have to do it like 3 or 4 times a week, okay? So it's really important aspect. Okay, I want to throw that in there. And then we get to the end. So now the collecting duct, right, collects all that urine, and it goes into the renal pelvis, and it gets into the ureters, right? And here our ureters are connecting to the urinary bladder, right? And we can store the urine there, and we store it there until enough pressure builds up, and it puts pressure on the internal sphincter, which is involuntarily controlled. So as that pressure causes simple reflex, causes the internal sphincter to relax, you go, oh, I have to go to the bathroom. But luckily you have an external sphincter, which is skeletal muscle, and it's under voluntary control, and you can clamp that baby shut so you get to the appropriate area to release that urine. You can't do it forever though. Remember, skeletal muscle fatigues, right? Can't do it forever, right? So here the next slide kind of shows the reflex. This is called voluntary and the reflex control of Micturition. Micturition is the action of urination, right? So that's everything I went through. The bladder fills, you've got stretch receptors in there. That can cause a couple different things going on here. You've got parasympathetic nerve, all right, acts on the bladder, causing the bladder to contract and causing the internal sphincter to open, and then your external sphincter will open too, right? And you get urination, okay? You can also, this can be overridden by the sympathetic. So the idea here is parasympathetic, rest and digest. Good time to go to the bathroom is when you're not in any danger. Think of yourself as a gazelle on the plains. If you're squatting, going to the bathroom, a lion can get you. So normally you want to do it when it's nice and calm. Sympathetic can override all of this and cause you to empty your bladder too. So we have dual innervation, because the idea is if you need to run from a predator, it's better to release the fluid in here so you're lighter and you can run faster. That's seriously the idea behind it, okay? But here's, again, voluntary control of the external sphincter, right? And so you can keep it closed and you don't have urination. Okay, that's it, guys. That's it for the urinary system. Thanks for listening.